What's going on, everyone? Welcome back to another phenomenal Biochem 2 video. Um, yeah, you guys have an exam this week. Your first Biochem 2 exam. And honestly, we covered a decent bit of information. I understand this is all new, or most of it's new. So today's video, on top of covering some of the newer content that we need to know, I'm also just going to be doing a huge review of past content as well. So I guess you can consider this video a crash course. I will be doing a crash course for Biochemistry 2 on Tuesday. So if you watch this video and you feel pretty comfortable with it, feel free just to do independent study and do some practice questions. But if you want me just to hear another run through again in person, feel free to join on Tuesday. Shoot me a text. I don't know for sure where I'm doing it yet, but I'll figure it out as we get closer. What else? I don't know, honestly. Again, going to be doing run through all the way from the top. So if you feel pretty good about all this content, feel free to fast forward into the newer stuff. If not, I'm going to be doing a lot of drawing this video. So I guess pull up the packet, follow along, and let's get into it. So if we remember, and I'm going from the very first page on this packet. So to start off, how do we get glucose into our body to begin with? We know glucose is involved in all these anaerobic, aerobic pathways in the body, but we'll do a quick recap on how it actually gets in. So, if we remember from the beginning, we will eat polysaccharides such as starch. And what our body is going to do is we're going to break starch down into individual components. So, on the left side here, we are going to have our individual components of maltose and maltotriose. And then over on the right side, we are going to have alpha limit dextrin. And if we go back to one of the earlier videos, our main enzyme here that's going to be breaking this down is alpha amylase. And alpha amylase here is supplied by saliva and our intestines. It's kind of like the first stage of breaking down a lot of these bonds and the polysaccharides. And if we remember, alpha amylase breaks down every other alpha 1,4 bond. So from starch here, as soon as starch gets into our body and our system, alpha amylase is going to act upon that starch and break down every other alpha 1,4 bond to give us maltose triose, and alpha limit dextrin. So these are, I just like to refer to them as our intermediate steps, but they can be broken down even more to get to our final level of glucose. And glucose is going to be one of our main components that gets absorbed into our blood. So if you guys see on a question or something, what is most likely to be found in the hepatic portal vein? Glucose is a great answer. Fructose is a great answer because, again, fructose is just an individual component. It's usually not a polysaccharide unless it's built in with a ton of other things. So just make sure to pay attention to some of the terminology that's used. But we're going to continue breaking down these intermediate components into glucose. So both of these intermediate steps gets broken down into glucose. However, these intermediate steps are acted on by different enzymes. So over here on the alpha limit dextrin, you're going to have oligosaccharidase acting upon alpha limit dextrin and alpha glucosidase. And then over here on the maltose and maltose triose side, pretty simple to remember, we have isomaltase interacting with maltose and maltose triose over there. Isomaltase over here on the left-hand side is going to be breaking down the rest of the alpha 4 bonds left over, but these two enzymes right here, they are acting upon every alpha 1,4 linkage as well as every alpha 1,6 linkage as well, and that is kind of specific to alpha limit dextrin. So just make sure to be able to differentiate what enzymes act on which bonds between our intermediate steps here. And another fun note to know is isomaltase and these two enzymes over here are both supplied by intestinal mucosal cells. Oh, did I spell that right? Yeah, if not, no worries, you guys understood what I'm saying. So intestinal mucosal cells supply both of these enzymes to break down our intermediate steps into glucose. And once we have it broken down into glucose, this glucose is going to come over here. So over here, we're going to talk about how glucose is absorbed from our intestinal lumen into our enterocytes or mucosal cells, and then eventually into our hepatic portal vein. And then from there, we can get into all of our fun pathways. So once glucose is broken down, we're going to be entering our enterocyte. And this is just going to be our enterocyte here. And we're going to have a channel protein right here, channel protein right here, and then over here on the far side, I apologize, I'm not that great at drawing, we are going to have our hepatic portal vein. And this is just going to be one of our main transports for glucose, for getting it into our blood, and then eventually can get passed down, broken down into all of our differentiating pathways. But glucose is going to start out in the intestinal lumen. We are then going to enter our enterocyte, or another name for it is our mucosal cell. These two terms can be used interchangeably, so make sure to have a good understanding of both, know what they mean, and I mean they mean the same exact thing, but you never know what question you're going to get. So we'll jump back up here, and the first channel protein we go through is called SGLT1 or 2. Both glucose goes through both of them, but again, we just need to know it's SGLT. It's an SGLT channel protein. It can be 1 or 2, and glucose is going to travel through it to get into our enterocyte. But it's also going to be paired with sodium. There's a huge concentration of glucose already in the enterocyte, and it's just kind of waiting to enter the hepatic portal vein. So we need a co-transporter, and that co-transporter is going to be our sodium ion right there. And it's just going to help glucose overcome that concentration gradient within the enterocyte to allow glucose to enter the enterocyte, enter the hepatic portal vein, and eventually go downstream. So once glucose has entered the enterocyte, it's going to continue passively through this next channel protein called GLUT2. GLUT2, just a big name we need to know just for getting glucose into the hepatic portal vein. GLUT2. And then from here, glucose will enter the hepatic portal vein, and as I said 200 times already before, going to go downstream to the through the hepatic portal vein into our later processes. And then sodium is also going to be inside this enterocyte as well. And the enterocyte, sodium is going to leave through just a little channel protein up top, but it's going to be paired with potassium coming in. So you could almost call this a sodium-potassium pump, but we really don't need to worry about that too much, and we just need to know ATP is going to be used at this pump up here because we need active transport to get sodium out of the cell. So it is going to use some ATP up here. In the bigger picture of things, it's just kind of a fun fact that we need to know. Sodium and potassium up there use an ATP, but downstream it really doesn't affect our overall ATP counts when we start getting into some questions. So we've covered how we get glucose into our body and into our bloodstream. So where do we go from here? Well, the first pathway we're going to be talking about is anaerobic glycolysis. So glucose, this is just going to act as our little bloodstream. Glucose is going to exit our bloodstream, turn into G6P. G6P will go to F6P, eventually to F16P, and from here it will differentiate into G3P, and then DHAP, and then, I do have more room. I used a smaller font so I can actually have full room to go through everything. Sorry, we are going to be going from G3P to G13P, and then we're just going to be getting into a ton of just conversions going downstream, but just for the sake of this video, I'm just going to shorten it up a little bit. We're going to go into, down here will eventually turn into phosphopyruvate, into pyruvate, and we'll end with lactate down here. And if we remember, anaerobic glycolysis does not involve the consumption of any oxygen. So if you don't see any oxygen talked about in any of the questions, you should know that you're in an anaerobic state. And anaerobic state has some individual little steps that we need to know, that's different from aerobic glycolysis. But biggest overarching concept here is our ATP count. So if we look right here, we can see again, we have phosphate. So we know right here under the enzyme hexokinase, we're going to be -1 ATP right here because we added a phosphate onto a glucose to make G6P. And then right here from F6P to F16P, we have the enzyme PFK. And if we remember that's an allosteric enzyme, it can be inhibited by products downstream. That is also going to be adding on another phosphate to F6P to make F16P. So in this first stage, we are going to be -2 ATP in this first stage. And then the divide for the second stage will kind of happen right here. So biggest rule that happens in the second stage is, again, everything happens twice. So G3P is the only product of F16P that can continue on to the second stage. We have DHAP right here, and DHAP is going to be used in instances where we don't need to generate any more ATP, or we just have an overabundance of products downstream. So DHAP is just almost an alternate pathway to store glucose, and it will mainly go towards adipose tissue. It will be used for fat storage, some fatty acid synthesis downstream for diglycerides and triglycerides. That is more of a minor detail. It's good to know what it does, but we will be getting into those concepts in the next exam content. But for what we really need to know now is the second stage of glycolysis. So second stage right here, our first entrance into that will be G3P. And we can see we go from G3P to G13P, which means we added on, and I always make this mistake, it's actually BP because it is a biphosphate, even right here, F16 biphosphate. Always forget about that. So G3P into G13 biphosphate, we know that we are getting actually, hang on a second. Yes, okay, sorry. I always get a little bit confused in some of the minor details that go into this, but G3P will eventually turn into G13 biphosphate. We'll continue down going to phosphopyruvate and pyruvate. And I'll keep it simple just to keep this video under wraps and not to get too long, but between phosphopyruvate and G13P, we go through a ton of different conversions. But what we need to know is after glyceraldehyde 1,3 biphosphate, we generate 1 ATP here. We take an ADP, generate an ATP, and so right now we are +1 in the second stage so far. And then from phosphopyruvate, we can see we have a phospho in the name, and we go to pyruvate, which signifies, again, we generate another ATP because we lose a phosphate. So ADP here, we turn into ATP, and again, we generate another phosphate or generate another ATP. And as I said before, everything in the second stage happens twice. So we don't only generate 2 ATP. Technically, in the second stage, we generate 4 ATP. But if we include everything in anaerobic glycolysis right now, our total net ATP count is only going to be +2 because we subtract the 2 ATP from the beginning, include the 4 ATP that we generate in the second stage, add them together, boom, net total of 2. And now we have some smaller steps that we need to talk about, specifically for anaerobic glycolysis. So when we go from G3P to G13 biphosphate, we're under the action of a dehydrogenase enzyme, specifically glyceraldehyde 3P dehydrogenase. And anytime you guys see dehydrogenase, you kind of want to assume an NAD or NADH is present because we are dropping off a hydrogen, we are either taken or given one, and that's kind of where the dehydrogenase term comes from. Furthermore, anytime you guys see kinase, similar to hexokinase right there, or phosphofructokinase, if you guys remember, kinase involves ATP, either making or breaking them, but it's kind of like a little cheat to understand how some of these enzymes, how their names go together with some of the processes that are going on. But from the beginning right here, we are going to be using an NADH plus to come up and interact with G3P here, and we are going to generate an NAD NADH. Furthermore, how we get that extra phosphate on there is we are also going to be having just a phosphate dropped off right there. And our product down here, along with NADH, is going to be a secondary hydrogen. I'd say the most important detail here is knowing how NAD plus kind of works with that, but understand that there's a phosphate that goes into it, and we also generate a hydrogen. Never a bad detail to know. And then fast forwarding downstream, over here on the pyruvate and lactate side, we need this NADH to be here to turn G3P into G13 biphosphate. So where do we get that from? And in anaerobic glycolysis specifically, we get it down here from the conversion of pyruvate into lactate. So basically what's going to happen is we're going to have an NADH come back, and we're going to interact with pyruvate here. We are going to reduce pyruvate into lactate because we are giving away a hydrogen atom. And as a result, NADH is going to be oxidized because it is losing something. Pyruvate is going to be reduced because it is gaining the hydrogen. And we're going to generate lactate as well as NAD+. And what's going to happen is this NAD+ is going to come all the way back to the beginning and jump right back in line and start the whole second stage all over again. And specifically for anaerobic glycolysis, this is really what we need to know. This is how we get that NAD8 or NAD plus back to the beginning to continue on this stage. And if you guys have watched my previous videos, we know that in aerobic glycolysis, it's a little bit different because we don't go from pyruvate into lactate. We stop at pyruvate in anaerobic glycolysis, but that is going to be a process I'm going to cover in a second. What else should I talk about? We do have a little bit of alternative pathway right here, if you guys remember. In instances of high elevation, we are not going to go from G3P into G13 biphosphate. We are going to turn into G23. Yeah, we're going to be turning into 2,3 biphosphoglycerate. And actually, we actually don't need a G there. I always just get these little minor details wrong, but don't worry about that. So up here, we're going to be turning into 2,3 biphosphoglycerate. And all this is just a secondary step. In areas of high elevation, our body needs to prioritize where our oxygenated blood is going to. So what happens is 2,3 biphosphoglycerate is just a competitor of oxygen. We compete for the binding sites on hemoglobin, which allows us to use our oxygen more efficiently and put it to the tissues that need it, and also reduce some of the oxygen that's going to some of these other tissues that may not be as important as some other areas inside our body. So that's just a little alternate pathway that we should know. It should always be involved with low oxygen areas. So if you're in Denver and you have a high elevation, this component is going to be activating up there. And then if you guys remember down here, we have our pentose phosphate pathway, also called the shunt pathway, also called the phosphogluconate pathway. And what we need is 3 G6Ps to get this guy going. And I did look up to make sure this pentose pathway, it's not specific to our anaerobic glycolysis. It can happen either or in aerobic glycolysis and anaerobic glycolysis. I'm just going to throw it in on this chart just so I can get it out of the way. But no, it's not specific to either of the processes. It's specific, similar to glycolysis, but not whether oxygen is involved or not. So from here in our pentose or shunt pathway, our main products down here is, I like to call it our intermediate product because it gets transformed into other stuff down the line, but Dr. G might use it as one of the main products. We're going to be generating ribulose 5P, and we're also going to be generating right here NADPH, and also a CO2. I'd say the most important products to know for this specific area is NADPH and ribulose 5P. And if you guys have seen my other videos, NADPH is not a redox agent. What NADPH's main role is to build macronutrients inside our body. So we will eat food, break it down into individual components, and when we need to make it into bigger structures like proteins, maybe fatty acids, whatever our body needs, NADPH is going to be one of our main components to build these macronutrients. But don't get it confused with NADH or FADH that we will talk about later, because those are specific to redox reagents or redox reactions. So again, going off the ribulose 5P, Our main products from ribulose are going to be two F6Ps as well as a G3P. So this is how the pentose shunt pathway is eventually incorporated back into glycolysis. And another little side note for this shunt pathway, ribulose is a pentose. So it has helped with the reconstruction of mRNA within inside our body, anything that involves any of our DNA. But to get it incorporated back into glycolysis, we are taking a pentose and we are putting it through some reactions to turn it into hexoses and trioses so it is compatible with the pathway. And again, there are some little bit deeper enzymes that are involved in the conversion of ribulose 5P into its products with transketolase and transaldolase. I'm not going to go super in depth with those on this. I'm just trying to give a huge overview on some of the systems that we need to know. And I believe for just anaerobic glycolysis, this should be pretty good. Minor details when it comes to the pentose phosphate pathway is this is present in erythrocytes and hepatocytes. So, aka red blood cells, aka liver cells, but it is not present in muscle cells. So when it comes to locations, again, these are huge locations to know. What cells have what function in pertinence to some of these processes that are going on. So pentose shunt pathway only happens in erythrocytes and hepatocytes. Furthermore, it does not use or consume any ATP, so it is not going to affect our overall ATP count that we already have going right here. Let me make sure I didn't miss anything, just so I don't have to come back and bore you guys to death with this first pathway again. I guess I'll just run through DHAP process again, just because I'm reading it right now in the packet. Increased glucose levels and decreased ATP demand will eventually result in adipose tissue, like I said earlier. From DHAP, it'll go through a series of reactions here, but let's say you eat a huge loaf of bread, you're not very active, and you're just kind of sitting on the couch. Well, glucose over here, we're going to start breaking glucose down through all these pathways, and it's going to get here, but we already have an overabundance of ATP downstream in this area because we're not being active. So what's going to happen is F16 biphosphate is going to turn into DHAP, and our body's just going to say, hey, let's store this glucose in some storage form. It's going to go through a ton of different processes to be turned into eventually glycerol and eventually into triglycerols and diglycerols downstream. Talked about that a second ago, but it's never a bad idea to know. But yeah, for anaerobic glycolysis, that is what we need to know right now. So let's get into the fun stuff when we talk about aerobic glycolysis. And again, for this now, since we covered a lot of the minor details up here, I'm just going to simplify our little roadmap here. I'm going to go glucose to G6P, then F6P, F1, 6BP, and then again, G3P, and then I'm just going to fast forward down here. Oh, actually, I'm going to shorten those arrows just so we have a little bit more room. We're going to fast forward down this way. We'll turn into pyruvate. And I'm just going to keep lactate on here just for the sake of it, just to keep our whole roadmap present. But we are in an aerobic state now, which means we are consuming oxygen and using CO2 inside our body. So a couple different things are going to happen on here in an aerobic state. If we remember again, right here is kind of the cutoff for going into the second stage. But just to fill in some minor information again, right here we are losing ATP. Right here we are losing an ATP because we're adding on phosphates to these products. And then right here we're going to be generating an ATP. Right here we're going to be generating an ATP. So if we just want to keep track of our ATP counts, again, first stage, which is the investing stage, we are going to be -2. Over here in our second stage, we are technically, specifically speaking for the second stage, since everything happens twice, we're technically generating 4 ATP, but when we subtract the 2 from the investing stage, we're left with a net total of plus 2 ATP for all these beginning stages here. Now, what's different in aerobic glycolysis is we do not go to lactate. We stop at pyruvate and put it through a couple different highways. So the different highways we're going to be talking about is the PDH system, and then eventually we are going to be talking about the TCA cycle down here. So I'm just going to throw oxaloacetate, and if I probably spelled that wrong, we're going to be talking about acetyl CoA, and then we're going to have citrate right here. And again, if we remember that citrate is made from the combination of acetyl CoA and oxaloacetate to make citrate, and then eventually citrate will go through all these different processes in the TCA cycle and generate a ton of energy for our body. But let's not get too ahead of ourselves. So I'm just going to draw these squiggly lines just to reference locations. If we remember over here, when we were talking about the PDH system, or the pyruvate dehydrogenase system, we are inside the mitochondrial membrane. So the PDH system is the connector from glycolysis into the TCA cycle. And basically what we were doing is converting pyruvate into acetyl-CoA. And there are a ton of vitamins and cofactors involved with the PDH system. I mean, we have vitamin B1, we have vitamin B5, I'm pretty sure, pantothenate. And that, if you guys remember, that's coenzyme A. So we have, to get this CoA down here on acetyl-CoA, as pyruvate's going through the PDH system, we are going to be adding on a CoA enzyme, or if you guys remember, I believe it's vitamin B5. Correct me if I'm wrong there. And then we are also sending in an NAD plus in here as well. And then as our products, we are going to be getting acetyl-CoA. We're going to be getting an NADH, and we're also going to be getting a CO2 out of it. And this whole series of reactions is acted on by pyruvate dehydrogenase. And if we remember, dehydrogenase, that's signifying that it is involving a hydrogen somehow. And in this instance, we're taking hydrogen off of pyruvate and converting it into an NADH, and then acetyl-CoA is going to be one of our products. And we also generate ATP in this step. So through a redox reaction, we are actually generating 2.5 ATP here. And what basically goes on is we consume half of an oxygen right here, and we produce an H2O molecule. So we consume half an oxygen, run it through with NADH, and as a result, we get 2.5 ATP from this reaction. But if we remember, everything in the second stage happens twice. So we don't only produce 2.5 ATP. In total, we produce 5 ATP here. So we did also talk about allosterically how pyruvate dehydrogenase gets shut off. And if you guys remember, we had PDH kinase here as a negative modulator for pyruvate dehydrogenase. So anytime we get an overabundance of acetyl-CoA or NADH, and by overabundance, I mean we are not using it for the TCA cycle because maybe we have too much energy already made, or we don't have enough oxaloacetate ready to combine to go into citrate. It's basically just a backlog here. Acetyl-CoA is getting accumulated right there. We have an accumulation of NADH, and what these two molecules are going to do is they're going to activate PDH kinase, and PDH kinase is going to come over here and inhibit pyruvate dehydrogenase, so we don't continue to break down pyruvate into an already overabundant population of acetyl-CoA. And then if we want to turn that enzyme back on, if you guys remember, we use PDH phosphatase. I'm pretty sure. Let me fact check myself here. Yes, PDH phosphatase. And that'll be a positive modulator for this enzyme. That means we are turning it back on and getting it kicking again and converting it allowing pyruvate dehydrogenase to convert pyruvate into acetyl-CoA. So for what we need to know, PDH system, generate 5 ATP total. It's within the mitochondrial membrane. We're producing acetyl-CoA and NADH, and we're also activating different enzymes on whether or not we need to inhibit or activate pyruvate dehydrogenase. And it is just a connector between glycolysis and the TCA cycle. So now let's talk about the TCA cycle. And again, oversimplified view. I'm just giving you guys the main concepts that we should know, and then all those minor details are going to be for you guys, because if I had to study them, you guys got to study them. I'm not going to give you guys an easy path here. So what starts the TCA cycle? Acetyl-CoA doesn't start it. Oxaloacetate doesn't start it. The only way the TCA cycle starts or activates is from the combination of acetyl-CoA and oxaloacetate. And we have a little enzyme right here called citrate synthase. And that enzyme is going to be activating the basically combining of oxaloacetate and acetyl-CoA to make citrate. And once we get citrate, the TCA cycle starts. So what big concepts do we need to know about the TCA cycle? Well, what we really need to know is we have 4 redox components involved with the TCA cycle. Per 1 turn, we consume 2 molecules of oxygen, and we also produce 2 molecules of CO2. And in total, per 1 turn, we generate 10 ATP. So let's break down this ATP count. We are going to be having 4 redox reactions, and then we are also going to be generating an ATP between, between succinyl-CoA and succinate, and we're just going to be generating a random ATP. So, succinyl-CoA into succinate right here. And right in this step, we're just going to be generating an ATP right there. And again, you guys see CoA, so we know that the B vitamin B5 is going to be involved there to add on that coenzyme A to succinyl in the previous steps right here. And we're going to be generating an ATP in between there. But we're also going to have 4 redox reagents. So we're going to have a redox reaction kind of going on over here. We are going to be having another one down here. We're going to be having another one up here. And then we're also going to be having another one in between malate and oxaloacetate right here. And all these redox reactions are consuming oxygen. We're producing H2O, and we're also producing some ATP along the way. So we are going to have 3 NADH redox reactions going on, and we're also going to have 1 FADH reaction going on. And if you guys remember, per 1 NADH redox reaction, we're going to be producing 2.5 ATP per reaction, and we're also going to be producing 1.5 ATP per FADH reaction. So if we just really want to break down a redox reaction again, we're going to have NADH here. One of our main products is going to be NAD+, and that NAD will go through a different reaction, pick up that hydrogen. I really don't know where from, whatever it's reacting with, but we're also going to be consuming half of an O2 right here, and we are going to be producing an H2O up here, and then we're also producing 2.5 ATP. But if we remember, everything in the second stage happens twice, so times that by 2, and we get 5 ATP present per 2 rotations of the TCA cycle. And then FADH is basically the same exact thing, except we are producing only 1.5 ATP. So this is kind of where oxygen consumption comes into play, because we have 4 redox reactions, which means we have 4 half oxygen molecules that we are consuming. So if we consume 4 half molecules of O2, technically we consume 2 molecules of O2, and then just out here in random steps, we're also just pumping out CO2 at different stages on the TCA cycle out here. And again, huge overview, not going to go into super deep detail, but in addition to producing 9 ATP from this redox reaction right here, from our redox reactions alone in the TCA cycle, and the addition of this ATP right here, we get 10 total ATP per 1 cycle of the TCA cycle, but everything in the second stage happens twice. So in reality, we are producing 20 ATP per 2 cycles. We're consuming 4 molecules of O2 per 2 cycles, and we're also producing 4 molecules of CO2 per 2 cycles. So, in reality, our main ATP count here is actually 20 ATP, and aerobic glycolysis actually produces 30 ATP per cycle. So where's this last 3 ATP coming from? Because we have 2 up here, we have 2 right here from our initial component of glycolysis, we have 5 ATP here present in the PDH system, and we also have 20 ATP down here present in the TCA cycle. So we're at a total of 27 moles of ATP. So where do we get this last 3? And this last 3 will come from what we call the glycerol P shuttle, or the glycerol phosphate shuttle. And if you guys remember from a second ago, how I was talking about how this NADH here is needed to kick off the second stage. In aerobic glycolysis, we don't go to lactate, so we can't oxidize NADH into NAD to restart this whole process again. And this is where the glycerol P shuttle comes into play, because we need to generate that NAD plus somehow. So G3P 1,3 biphosphoglycerate, and then just a huge jump down here to pyruvate, and then over to lactate right here. So again, this is strictly for aerobic glycolysis. And basically what's going to happen is we can get all the way down to pyruvate, and we'll go through the PDH system down here and eventually the TCA cycle down here, but we're going to have an accumulation of NADH right here because it is used to being reacting with pyruvate to produce lactate, but since we don't go to lactate, we can't use it. So we're going to be going through the glycerol phosphate shuttle here, and we know over here we need this NAD plus here to eventually react and turn into NADH. But how does it get there? So we're going to have a series of reactions going on in the glycerol phosphate shuttle. We're going to have DHAP reacting right here, turning it into glycerol P. Glycerol P is going to be down here and is going to react with FAD plus to produce FADH. FADH is going to come down here, and if you guys understand, we are in an aerobic state here, so we are going to be having a redox reaction. So down here, we're going to be reacting with half of an O2 to produce H2O. So how does NADH all the way over here fit in with all of this? Well, this NADH is going to come flying in here, and it is going to give off a hydrogen to DHAP to turn into glycerol P, and that free-floating NADH is going to come right back up here and kick off the second stage all over again. And glycerol P is going to come down here, react with FADH. FADH is going to become reduced because it has the hydrogen right there, and I know in the text it says FADH2, so whatever floats your boat there, I'm just oversimplifying it here. But what we really need to know is, since we are consuming O2 and having a redox reaction, we're going to be producing ATP right here. And per 1 FADH, we produce 1.5 ATP. But I hope you guys caught me there. We're in the second stage, so we don't only produce 1.5 ATP, we technically produce 3 ATP because everything in the second stage happens twice. So if we fit all this together and do a quick overview again of aerobic glycolysis, first stage, investing stage, minus 2 ATP. Then we go into the second stage of glycolysis and we produce a total of 4 ATP. But in this whole long chain right here, technically, we're only net 2 because we subtract the 2 investing stage ATP from the total of 4, get plus 2. Then pyruvate goes through the PDH system here and we produce a total of 5 ATP per 2 cycles. And then eventually we get down here into the TCA cycle, produce 20 ATP, and lastly down here we produce 3 ATP through aerobic glycolysis. So if we want to do a breakdown, glycolysis is equal to 2 ATP, then we have PDH, which produces 5 ATP, and then we have TCA cycle, which produces 20 ATP, and then we have the glycerol P shuttle, which produces 3 ATP. So as a result, we produce 30 ATP. And there we go. That is the aerobic glycolysis broken down as a huge long review again. And right up here again, aerobic glycolysis with only a net total of 2 ATP. And then we also have our beginning stages up here. So with all this out of the way, we can get into the new content now. So where to start? Where to start? We are going to start with glycogen. So if we go all the way down here, right here, glycogen. So glycogen is a homopolymer of glucose. So basically it is just a long chain of glucose. And we have two main areas that we store glycogen within the body. We have the liver and we also have skeletal muscle. Skeletal muscle will contain the greatest source of glycogen in our body. And kind of how it works is glycogen is just a way for our muscles to store energy or to store glucose. So realistically, the main example you guys have probably heard is if you are a marathon runner, the night before you're eating a huge carb-loaded diet so you can give an abundance of glucose into the body and eventually have it be put into storage, because if you're resting, you're not going to be using it. So let's say you eat 5 different plates of Olive Garden, get all in on those breadsticks, some of the fettuccine Alfredo, something like that. Glucose is going to be entering our body here. Oh, wrong signifier right there. Let's see if I can, yeah, whatever. We're going to be eating glucose right here, and we're going to get to G6P, but we're not active, and we don't really need an ATP count downstream yet because we're not running. So what our body's going to do is through the enzyme phosphoglucomutase, we're going to be taking G6P, converting it into G1P, and then using the enzyme glycogen synthase to eventually produce glycogen. And what we need to know in reference to ATP is it takes 2 ATP to convert glucose right here into glycogen all the way down here. But to go from glycogen back to glucose, it does not consume any ATP. It's a pretty standalone process. And once we get our glycogen convert or our glucose eventually turned into glycogen while we're sleeping, and let's say we start the marathon the next day and we're running a ton, and some of maybe the big breakfast you guys ate, we used up all of our energy within the first 5 to 6 miles. So now our body's going to be looking to alternative sources of energy to keep these muscles firing, to keep ATP going, to keep our body energized. So when we get to that 5 or 6 mile mark, our body's going to tell our glycogen storages inside our muscle cells to go, hey, we need some ATP. We need to have glycogen get converted back into G6P to eventually go all the way back down to the TCA cycle. And if you guys remember, all of those processes that eventually get to the TCA cycle are huge components of making ATP within inside our body. So when our body tells us, hey, we need energy, our body's going to take our glycogen storages here and we're going to throw through the process of glycogen phosphorylase. We're going to pair it with a phosphate right here to throw that phosphate group to have glycogen turn into G1P. And then we are going to be converting G1P back into G6P and send it down the way towards the TCA cycle so we can continue to break down that G6P and eventually create energy for our body. Now, this is highly specific to skeletal muscle because we are breaking down glycogen into G6P to put it towards the TCA cycle to generate energy. Now, you guys see the liver up there on the screen right here, and the liver kind of has a different process when it comes to glycogen. When it comes to glycogen, we're kind of going to be more modifying our blood sugar levels rather than generating energy. We are going to be turning glycogen back into glucose so we can either put it back into our blood to modify our blood sugar levels. So like with people who are diabetics, insulin, which we will talk about way downstream, I believe exam 3 content. When our body tells us like, hey, we need to raise our blood sugar level, what the liver is going to do is it's going to take the glycogen storage that it has within the liver. We're going to turn glycogen again, same process, back into G1P with the addition of a phosphate right here. And when we get to G1P, we're going to turn it back into G6P here. But instead of going down the way towards the TCA cycle, we are going to use an enzyme called glucose 6-phosphatase. And basically what's going to happen is instead of going this way over here, our body's going to say, nope, we don't need to generate any energy. We need actual glucose back in our blood to modify our blood sugar levels or to maybe make some other products downstream. But we're going to use the enzyme glucose 6 phosphatase right here. And this enzyme is highly specific to the liver. The liver is one of the few areas that does this in the body that allows us to convert products of glucose back into glucose. So we're going to take G6P here. We're going to take glycogen again, turn it into G1P, put it back up here to G6P, and through the activation of the enzyme glucose 6 phosphatase, it allows us to turn G6P back into glucose here to put back into our blood or to send down some alternate pathways. So if I just do a huge, quick, simple overview to kind of make this pretty simple, if we need energy inside our body, like we're a marathon runner and we need some extra energy for our muscle cells, we convert glycogen into G1P and then G1P into G6P, and then we'll send it down the way to the TCA cycle to generate energy. And if we need to modify our blood sugar levels through the use of our liver glycogen levels, we're going to convert glycogen into G1P, convert it back into G6P, and then through the enzyme glucose 6 phosphatase, turn G6P back into glucose to put back into our blood. And then we do have just some smaller fun facts that we need to talk about right here. So the liver can also break down glucose as well and turn it into G6P, kind of like in a similar example up here. So instead of going back from G6P to glucose, we're going to go from glucose to G6P, and then eventually downstream, TCA, PDH, whatever process we need to run. But if we're in the liver, we use a different enzyme right here to convert glucose into G6P. So if we're in the liver, we use glucokinase to convert glucose into G6P. And again, we are using an ATP here very similar to hexokinase. And then if we are in skeletal muscle, we are using hexokinase instead to, again, add a phosphate on to G6P. So liver, glucokinase, skeletal muscle, hexokinase. Be able to differentiate between the two. If he says we're in the liver, something like that, no, you are using the enzyme glucokinase. And if we're in skeletal muscle, no, you're using hexokinase. And then just some terminology here, glycogenesis, glycogen synthesis, glycogenolysis, breakdown of glycogen to glucose. What else do we need to cover here? Well, let's just talk about the fructose pathway. So if you guys remember back from Biochem 1, fructose is found in honey, cane sugars, and high fructose corn syrups. And if you guys remember again, honey, mixture of fructose and some other things. High fructose corn syrup, again, is a mixture. So we do not need to digest these. We do not need to digest these mixtures because our monomers are already free floating there. We can just absorb them into our body. Now, if we have sucrose instead, sucrose needs to be broken down because it is a polysaccharide, and we have to get those bonds broken to expose or isolate those monomers that are present. And just another fun fact to know, the total ATP produced from fructose is 30 ATP, so it's very similar to breaking down glucose in the aerobic process. Fructose is very similar in that way because, again, fructose is another monomer, very similar to glucose. But the fructose pathway is a little bit different. So for the fructose pathway, we start off actually in the liver when we're breaking down fructose. And from what you guys can see right here, one of the main enzymes that's acting on fructose to turn fructose right here into its product, which is going to be fructose 1P. So we can see that the first enzyme to turn fructose into fructose 1P is fructokinase. And again, anytime you guys see kinase, I want you guys to be thinking about ATP. So to turn fructose into fructose 1P, we can see that we add a phosphate. So that means we are spending an ATP to add a phosphate onto fructose to eventually turn it into fructose 1P. And then from fructose 1P, if we want to break it down even further, we can break it down into either DHAP or we can go all the way to glyceraldehyde 3P. And the main hiccup, or oh man, I'm having a brain fart right now on the right terminology to use. Almost, I'm just going to say gatekeeper. The main gatekeeper of turning fructose 1P into DHAP or glyceraldehyde 3P is going to be aldolase B. Aldolase B is going to be one of the main enzymes that acts upon fructose 1P. And if you guys go and look at some of the practice problems that are floating around, we can see that a lot of the questions that reference fructose is going to reference the enzyme aldolase B. So if someone has like a fructose intolerance, you can almost guarantee that the enzyme aldolase B is either not functioning properly in their body, or they just don't have a lot of aldolase B. So if aldolase B does not work here to turn fructose 1P into our products down here, we're going to start to develop an overaccumulation of fructose 1P. And this overaccumulation actually is not that great for our body. And if we flash forward down here, I apologize, I just lost my train of thought for a second. Fructose intolerance due to an aldolase B deficiency, which means there is an abundance of fructose 1P in the cells. This is genetic, and furthermore, hepatocyte death occurs from an overaccumulation of F16P in the cells because we cannot phosphorylate this free-floating phosphate around on F1P downstream because this enzyme right here is not working. So this phosphate is just going to be stuck here on fructose 1P. But let's say aldolase B, we don't have a fructose intolerance. Let's say our aldolase B is functioning properly. We can turn it into either DHAP, which can go back to gluconeogenesis, which is the production of glucose from alternate sources within our body, or we can put it towards glycolysis, which is the breakdown of glucose within our body, or the breakdown of fructose technically within our body. Fructose, we put down the same pathway as glycolysis, just at a different point. So we know here going from fructose to fructose 1P, we use 1-ATP with the enzyme fructokinase to get our product fructose 1P, and then fructose 1P will eventually go through aldolase B. Now, if it stops right here at DHAP, it's going to go through an alternate pathway, but it really doesn't involve any ATP. If we want to break down fructose even further to put down on the pathways to generate some energy for ourselves, we're going to go through the enzyme triose kinase right here, and we're going to be using a secondary ATP to we're going to invest another ATP to eventually turn fructose 1P into glyceraldehyde 3P. And so if you really want to look at it, inside the liver, this stage of fructose catabolism is very similar to glycolysis in the fact that we invest 2 ATP in the initial stage to get to our end component of G3P to enter the secondary stage. And that's kind of where the ATP counts are very similar, because we're using 2 ATP in the beginning stage, and then we're generating all this ATP downstage. And I guess for fructose, I mean, I'll go down and explain this little pathway right here just so we know directionally where some of the stuff is going. But we also need to know some of the different specialized cells within inside our body. So, and the main differences between a lot of these different cells is whether or not it participates in anaerobic respiration or aerobic respiration. And it's very similar just to an anaerobic glycolysis or an aerobic glycolysis. So what we need to know is our red blood cells here only participate in anaerobic respiration because there are no mitochondria present in red blood cells. So that means no TCA and no conversion of acetyl-CoA via pyruvate dehydrogenase. Then when we talk about muscle and liver cells, they can function in both anaerobic or aerobic respiration depending on the state of the body and the needs of the body. And then lastly, when it comes to brain tissue, CNS neurons and stuff like that, it only participates in aerobic respiration. So being able to differentiate what cells participate in different pathways is a huge need to know for your upcoming exam. And then we covered this. I'll come back to this chart right there. We'll talk about adipose tissue real quick. So adipose tissue and glycolysis, it converts glucose to acetyl-CoA, but it is converted to fat for storage instead. It does not go through the TCA cycle. It does not combine with oxaloacetate to kick off the TCA cycle. It goes through a whole separate process that we'll get to in the second exam material. But for now, you guys just need to know any acetyl-CoAs generated kind of from adipose tissue is not used for the TCA cycle. Mano stuff right here, don't remember seeing a whole ton about that, but again, please go and look for the practice problems and see if any of that's referenced, because you are going to learn a lot more from doing a lot of this stuff on your own compared to just watching me and expecting to memorize it. You guys need to get the repetitions in to get this knowledge inside your head. So we'll talk about this chart real quick, and this chart is just a comparison between the pathway of glycolysis and fructose pathway. So we can see we have glucose right here, and again, we progress through all these different stages down here to eventually convert glucose down into pyruvate, and pyruvate can go off and do whatever it does, whether or not you're in aerobic or aerobic process, but fructose is also needed in here. And we know that fructose 1P can be converted into DHAP or glyceraldehyde, and eventually into glyceraldehyde 3P, and these two molecules kind of act for two different processes. So for gluconeogenesis, which is basically taking components of that inside our body and coming back up here to either produce glucose or produce glycogen, DHAP is going to be one of our main molecules for that. So we're going to break down fructose 1P into DHAP, and from here, DHAP is going to kind of go through the process of gluconeogenesis and go back upstream through fructose 1P biphosphate, back into fructose F6P, and eventually up here all the way back into glucose 6P, which can either be transformed into glycogen, go to glucose, basically whatever the body needs. This concept is going to be more important in later exams, but for now I just kind of want to give you guys more of a direction on how some of this stuff goes. But let's say we don't go to DHAP and our body needs energy and we want to break down fructose for energy. Well, we're going to go through the whole same thing again. We're going to go fructose, acted upon by fructokinase into fructose 1P, and eventually we're going to be acted upon by aldolase B to turn into glyceraldehyde right here, and this glyceraldehyde is going to be acted upon by triose kinase again and be turned into glyceraldehyde 3P. And what do you know? Glyceraldehyde 3P continues on the same exact way as our glycolytic pathway, and it's eventually turned into pyruvate, and from here it can either go to, aerobic glycolysis through PDH and the TCA cycle, or it can go over here and just go straight to lactate. You never know. It just depends on what the body actually needs. A lot of this stuff right here is just more terminology than anything. So gluconeogenesis is just the formation of glucose 6P from non-carbohydrate sources. So that's kind of what we saw right here with fructose, with it producing DHAP to come all the way back up here to form G6P. And non-carbohydrate sources, which means none of the ingested glucose or carbs that we get is being used for gluconeogenesis. It comes from other factors within inside the body. And the liver is the most important in the conversion of glycogen to glucose via glycolysis, which, again, as I referenced before with diabetics, it helps increase blood sugar. And again, glucose is the only tissue that can convert glucose 6P back into glucose. Muscle cells does not do that. Glucose is the only one because it contains the enzyme glucose 6-phosphatase to turn G6P back into glucose. But all skeletal muscles is just going to turn glycogen back into G6P and send it downstream towards the TCA cycle when it's needed. And then I guess right here, this is just going to be a ton of a review of the inhibition of glycolytic pathway enzymes. So these are great to know because realistically, if you view this like a highway, if there's a backup downstream, of course, we're not going to want to break down any previous molecules, because if we already have an overabundance, we really don't want to add to all that. So this is kind of playing on the role of allosteric enzymes being inhibited by their products. So when we talk about hexokinase is, again, the conversion of glucose to G6P, and an abundance and overabundance of glucose 6P in muscle cells is going to inhibit hexokinase, because, again, if we produce a ton of G6P that we're not breaking down into F6P or F16 biphosphate, why do we want to break down any more glucose? So glucose 6P, the overabundance, is just going to come back and shut down hexokinase, so we don't break down any more glucose. We also talk about phosphofructokinase, which is stimulated by the presence of AMP and a shortage of fructose 1,6 biphosphate. So this tends to occur during contracting muscles because we are using ATP, we're kind of producing AMP, and we are using up our fructose 1,6 biphosphate storages. So as soon as those levels drop, we're going to activate PFK again to convert F6P into F16 biphosphate so we can continue producing energy for the body. But PFK is also inhibited by high levels of ATP and citrate. So if we remember downstream, Citrate's one of our starting components for the TCA cycle. So if we break down all these components of glucose and we get down to citrate, and the TCA cycle doesn't use citrate, or we just have an overabundance of it because we're sitting on our couch and we're not needing the production of energy, those high levels of citrate and ATP that we make along the way are going to come back and inhibit PFK because we don't need to break down anything else out of the first stage. We don't need any more energy, and we just don't want to have an overabundance of all these products that we're not going to use in the moment. And that usually just occurs during resting muscles, which makes sense because we're not doing anything. We don't need energy. We'll talk about pyruvate kinase, which is inhibited by the activation of protein kinase A. Actually, protein kinase A, because pyruvate kinase, again, if you guys remember, is the conversion of phosphopyruvate into pyruvate, and it helps produce ATP there, and it's inhibited by protein kinase A. But the information about adenyl cyclase and protein kinase A, that is content for exam 3. Learn it if you feel inspired, but it's not a need to know at this instance. It's going to be a much heavier topic in later exams. Again, we have pyruvate dehydrogenase coming up. And you guys remember pyruvate dehydrogenase is in between pyruvate and acetyl-CoA, which is one of the products. And a high abundance of acetyl-CoA and NADH are going to act as an inhibitor for the PDH system, or pyruvate dehydrogenase. And it's a very similar concept to what we've been talking about for all these previous enzymes. If we are producing a ton of acetyl-CoA and a ton of NADHs, but we're not using them, we don't want to continue breaking down pyruvate dehydrogenase. We don't want to continue breaking down pyruvate. We want to take that pyruvate and maybe put it through some other processes to where it can be used, but we just don't need to use it for energy production. So a high abundance of acetyl-CoA and NADH that's not being used for the TCA cycle is going to stimulate PDH kinase, which again is going to be a negative modulator for pyruvate dehydrogenase, and it's going to shut it down, and the pyruvate that's floating around up there is going to be put through some other processes within inside the body. And then let's say our levels balance out again, we start running, we need some energy. PDH phosphatase is going to be one of the enzymes that comes back and activates pyruvate dehydrogenase. And again, this is stimulated by insulin and increased calcium 2+ levels. And basically the whole insulin move is when our pancreas secretes insulin, it's basically telling us that we have a high glucose content within our blood. So if we have a ton of glucose in our blood, we want to start pulling that glucose out and putting it through glycolysis to break it down to decrease the glucose levels within our blood. So by activating pyruvate dehydrogenase, all of those downstream products from the glucose that we're breaking down can be put through the PDH system, can be put through the TCA cycle and broken down for energy and all these other components. But the main need is just to get that main glucose out of our blood. And again, right here, isocitrate dehydrogenase. This is an enzyme within the TCA cycle, and alpha-ketoglutarate dehydrogenase. Again, both of these are components of the TCA cycle. And as I said before, I'm just not going to spit enzymes at you and expect you guys to know them right off the bat. That is just going to be something you guys have to do on your own. Repetition is the biggest thing, but isocitrate dehydrogenase is, again, stimulated by an abundance of ADP, which means we don't have a lot of ATP generated. We need to start generating ATP. So of course, isocitrate dehydrogenase is going to turn back on by a high abundance of ADP because we want to continue that TCA cycle to run to produce ATP. And it's going to be inhibited by an abundance of ATP and NADH. And again, same concept. We have too much ATP that we're not using, and we have too much NADH that we're not putting through redox reactions to convert energy. So if we just have an overabundance of this, isocitrate dehydrogenase is going to be shut down. And then furthermore, an abundance of citrate inhibits citrate synthesis, which again makes sense because if we're not using it, it's going to shut down the enzyme that makes it. And again, right here, alpha-ketoglutarate dehydrogenase inhibited by abundance of GTP, succinyl-CoA, and NADH, which these are just products of alpha-ketoglutarate dehydrogenase from turning, I believe it's alpha-ketoglutarate into succinyl-CoA. So an overabundance of that is going to shut down that enzyme. And then an abundance of calcium 2+ ions or a lack of succinyl-CoA and NADH is going to activate this enzyme just so we can keep that TCA cycle going. But this is probably going to be a long video, but I'm glad I was able to go through everything. This is just a huge crash course in everything we need to know for the TCA cycle, and then I am going to lock down a day to do a huge crash course again. So if you guys are confused on some subjects I covered today, feel free to come to the crash course and we can go through it all again. But I hope you guys had fun. I had fun talking about it. So start hitting the books. I know you guys have a loaded exam week, so I wish you guys the best of luck and good luck. So signing off. Have a great one.